obviously it's from because I've known the patients now also for some time, and in that respect, it's it's a very dramatic disease. It gets to you in a way that you see how the people who are affected by it and also the family around it are so much struck by this disease and how it affects their lives. So in that respect, but that I came to know after I got into Barth syndrome research. So at that time, it was a very interesting subject, which was the product of a crucial discovery, which was a logical project. So it was okay. Now I'm going to do that. So nothing of what I've seen or what I feel now for the Barth syndrome foundation and the disease itself. Only the doctors meet the patients, not the researchers. The researchers get like blood or tissue or whatever, but not the meeting the patient itself. So and now you can see a lot of them, and you see them all different ages, growing into adulthood. So that gives it an extra dimension. The research itself also is very interesting because it turned to cardiolipin, a lipid which was a minor lipid, not very interesting, and that turned it all around to a very interesting lipid. And lipids are, in science, are really booming, so to speak, especially cardiolipin. So it's in that respect, it became a more and more interesting subject, and had to have a human disease related to such a deficiency is something that always boosts interest and relevance for this kind of research. Well, cardiolipin is also involved in the process of cell death, and actually it's programmed cell death. And that is something that is highly researched, also in terms of cancer. Because if you can modify or at least control this, you can possibly also, in a controlled way, let cancer cells die. And that would be very nice. You could send them a signal, okay, die, and then specifically these cells, and then they will run that program and do that. So that's why programmed cell death and cardiolipin plays a very important role in that path is very interesting. The fact that cardiolipin is involved in such a important and interesting subject as programmed cell death makes it, and it is involved in a human disease which affects its synthesis and generation of correct cardiolipin. That only makes it even more interesting. And in its own right, Barth syndrome is very interesting from a research point of view. And if you want to know about cardiolipin, you also want to know how it is made and how it's synthesized, because then also you may get pointers who will give you an idea of how to control it via control cell death or whatever. If you see the amount of publications that are out there or being produced each year, it's a line going up. So in that respect, the Barth syndrome Foundation have done excellent work of stimulating the already interest that was there by giving out grants and by having these kind of meetings. So, but there's absolutely interest, more interest than there was. The science progression is also judging from the publication is going much faster and faster. So in that respect, we are advancing in our knowledge on Barth syndrome, and the chance that you will then hit something that will become relevant for patient care itself is only getting larger. But at this time, the research that is being done is it's not ameliorating the patient care itself, except for the diagnosis part, which has now become much more easier. For instance, the blood spot test is a good example of making it easier for doctors to say, okay, let's check for Barth syndrome, and that's getting to, well, identifying the patient itself is already a very major step forward. So the test itself is really simple. You put a blood drop on a paper, and you can dry it and then send it to our lab, and we punch out a spot, part of this blood spot, and then do an analysis on it, which is very simple. And the advance is that you now don't have to send a whole lot of blood, which you have to draw from usually a very small child, which you don't have a lot of material on, so a blood spot is less is better in this. And you avoid costs, which are always a high threshold, even for developed countries as the United States, for insurances and so on, to get the test to us so we can do the test. This test is very important since it lowers all these thresholds, which makes it easier for a doctor, if he is aware of Barth syndrome, to say, okay, let's test for this disease and see if this is Barth syndrome. It's not an expensive test. It's easy to perform. Result is quickly there. Why not do it? And then you will get a higher detection level, and that's progress. You see the persons that are affected by the disease you're studying, and well, that gives you an extra boost as okay to do more and to want to do more, because well you see sometimes the hardship that is caused by this disease. Also see the listserv or mails, and then if you read that one of the patients has died, and you also remember him from the last conference, then that's a large impact. So it makes it more relevant. If you have if your test if the test that we've developed, or the repository that has been installed, or the grants that the Barth Syndrome Foundation has put out there, makes that there are more patients that are going to be discovered, better treated, research will progress faster. That's well a very big accomplishment, and something that I think would have been in a less advanced stage if the Barth Syndrome Foundation wouldn't have been here to catalyze that.